infections. Neutrophil development in the bone marrow takes
Okay, so we were talking about basophils. Okay, histamine and heparin. Remember,
approximately
histamine
10 days.
is one of those things causes
Mature
swelling
neutrophils are
with
released into the
allergens
blood
or other encounters that the body has with foreign substances. Basophils. All right, see the basophil cells here in the center. They stain due to basic dye. Okay, and they are found here in the blood. Okay, see the nucleus of the basophil, this area right in here. Okay,
where they
the whole
survive for
basophil
only
cells
3 to
stain
6
there,
hours.
and we see the cytoplasmic granules.
Because mature
These little
neutrophils
dots, okay, are the granules inside there. So the red blood cells surrounding it, and a few platelets also. Okay, white blood cells, eosinophil cells. They account for 1 to 4% of white blood cells, so quite a bit more numerous than your basophils that are only 0.5%. Eosinophils have red staining bilobe nuclei connected with a broad band of nuclear material. Okay. They also have a red to crimson acidophilic course.
are so short-lived.
Lysosome,
The bone marrow constantly produces large numbers of
granules, and they
neutrophils
lessen the
to meet
severity
the
of
body's
allergies
needs.
by reducing inflammation and lead the body's counterattack against
Our body makes natural proteins
parasitic
that can
worms.
signal.
Okay, eosinophils, okay, are these right here. You need to go back outside with Mommy, okay? While Daddy finishes.
Mommy said I could come here.
I know. It'll just be a few more minutes. I'm sorry, Daphne. All right. So here we see the eosinophil cells, and we have the nucleus of the eosinophils,
The stem
cytoplasmic
cell
granules,
to start
and
making
then you
cells
see the
that will
eosinophil
eventually
is
develop as
surrounded by
neutrophils.
red blood cells. Okay, agranulocytes include lymphocytes and monocytes that
One of the
lack
important
visible
proteins in this
cytoplasmic
process
granules
is called
and are similar structurally but are functionally distinct and unrelated cell types. They have spherical lymphocytes or kidney-shaped
granulocyte colony
monocyte
stimulating
nuclei.
factor, or GCS.
Lymphocytes account for 25% or more of the white blood cells, and they have large, dark purple, circular nuclei
The
with a thin
GCSF
rim
protein.
of blue cytoplasm. They are found basically enmeshed in the lymphoid tissue. Hard to distinguish them. They're in there, and they're not coming out. There are 2 different types of the lymphocytes, the T cells and the B cells. The B lymphocytes are stimulated by bacteria or toxins and give rise to plasma cells which produce antibodies. The T cells protect against viruses and other intracellular microorganisms.
Can work on the cells in
and
the
they
bone
attack
marrow
and destroy
that eventually
the
become
cells that are
neutrophils
infected,
and
the killer cells. Okay, lymphocyte. Okay, the lymphocytes right here, we see the lymphocyte in the middle. They're agranulocytes. Okay, the nucleus
signal
of
that
the
they should start
lymphocyte
becoming
and then the
neutrophils.
erythrocytes surrounding it.
The
The nucleus takes up most of the cell of a lymphocyte. Monocytes account for
Entire process by which stem cells
4 to
develop into
8% of total leukocytes. They are the largest of the white blood cells, and they have an abundant pale blue cytoplasm. They also have purple staining U or kidney-shaped nuclei. We'll see those in just a second. And they happen to leave the
Neutrophils
circulation of the
can take
blood,
about 10
enter the
days,
tissue,
and
and then they
includes
differentiate into macrophages. Macrophages are monocytes that were once circulating in the blood. Okay, they're highly mobile and are actively phagocytic.
During the first phase, the stem
They
cells and
activate
their offspring
lymphocytes
can
and mount an immune response. Okay, here we see the monocytes. Okay, the nice little
divide as they
U-shaped
develop and
nuclei,
mature.
very large,
This
but
step
very
takes about
U-shaped or
6
very
days.
kidney-shaped, depending upon the actual individual cell that you look at. Okay, here we see the platelets and red blood cells surrounding neutrophils and lymphocytes. Neutrophils and lymphocytes are very easy to differentiate. Lymphocyte is mostly nucleus, where we see this nice lobed U-shape or kind of curly Q-shaped neutrophil nucleus. Identifying white blood cells.
Then,
I got a
over another
video
4
on
to
that,
5
but my
days,
internet
the cells
is
mature.
not working at its highest. I want you to watch that. There's a link there on the PowerPoint for you to download, and that'll send you to a really good video there. Okay, platelets. There's also another video here So make a note of it.
Further,
matter of
divide.
fact, question 1 for this lecture. Okay. Describe
The mature neutrophils are
one
finally
thing from that
released into the
video.
blood.
Alright. Megakaryocytes. Megakaryocytes are the initial cells that cause the production of platelets. Okay? Preventing blood loss. Without platelets, we would not be able to prevent blood loss very easily. A series of reactions that help bleeding to stop. 3 phases will occur in rapid sequence. Vascular spasms that are immediate,
can fight bacteria and
vasoconstriction
protect
to
the body
help
from
slow
infection.
down the movement of that blood and to keep it from flowing. Okay, thromboxanes and endothelin can then cause the vascular spasms, and then you have the platelets come in and start to form together and get really bunched up because they can't really slide past. Platelet plug formation. And then eventually on top of that, you have more things coming in, the complement system, and that causes coagulation. Okay, platelet plugs. Platelets do not stick together or to other blood vessels at all. Upon damage to blood vessel endothelium, okay, with the help of the von Willebrand factor, will adhere to not other platelets, but to collagen. Okay, and they're stimulated by and then released from thromboxane and ADP, adenosine diphosphate. Okay, which attract still more platelets, and they stick to exposed collagen fibers and form that platelet plug. The platelet plug is then limited to the immediate
white blood
area
cell
by prostacyclin, and it can cause
are an important part
nice
of our
little
body's
blood
immune
clots,
system.
and it can seal up
Neutrophils
small breaks in blood vessels that occur many times each day. Platelet plug formation. So platelet adhesion occurs when von Willebrand factor connects
are a
collagen
special
and the
group of
platelets
white blood
together.
cells
The
that play
platelets
an
then
extremely
release,
important
and then the ADP, thromboxanes, and other chemicals activate other platelets. The platelets then aggregate, and then they bind to fibrinogen, connecting the platelets to each other, and this platelet plug is formed by accumulating
role in protecting the body
masses
against infections.
of platelets.
Mature neutrophils
Okay. Blood clotting or coagulation, okay, is the formation of a clot or a network of protein fibers called fibrin. Okay, blood clotting begins with
divide.
extrinsic or
New
intrinsic
neutrophils are
pathways.
constantly formed in
Both
the bone.
pathways end with the production of factor X. Factor X is caused by the extrinsic pathway, begins with the release of thromboplastid from damaged tissue. Intrinsic pathway begins with the activation of factor 12. Okay. Activated factor 10 and factor 5, phospholipids and calcium from prothrombinase. Prothrombin is converted to thrombin by prothrombinase. Fibrinogen
Marrow will be developed
is
and
converted to
mature.
fibrin
Mature
by
neutrophils
thrombin. Okay, insoluble fibrin
are
strands
released into the
form
blood,
the
where they
structural
circulate
basis
for
of clot,
3 to
and
12
fibrin
hours.
causes the plasma to become the gel-like trap. Fibrin then, in the presence of calcium ions, activates factor
and then
13.
move to other tissues, where they survive
Okay.
only 2 to
And then it causes cross-links in fibrin, strengthens and then stabilizes that clot. Away from the site of the injury, anticoagulants in the blood, such as antithrombin and heparin, help to prevent clot formation.
3 days.
So we
These
see
mature
the pathway
neutrophils
here.
can be
We see the
found in
extrinsic
large-
pathway up top and the common pathway at the bottom. The extrinsic pathway starts with thromboplastin, okay, which
and
is released
small blood
outside the
vessels,
plasma
as well
in
as in the
damaged
tiny
tissue.
capillaries.
Then the intrinsic pathway starts when that factor 12, which is in the plasma, is activated by coming
In
into
different
contact
tissues.
with the damaged
In the
vessel.
bloodstream, neutrophils act
Factor 12 right here.
as surveillance cells, searching for infections.
Whoops. Alright, so activation of that intrinsic or extrinsic pathway results in the production of activated factor X, factor 10. Okay, which is the beginning of the common pathway. So once factor X is activated, that is now the common pathway, which means the common pathway comes from both the extrinsic pathway and the intrinsic pathway, and once you get to the point in which factor X is activated, they're both the same. It's the common pathway. Okay. Activated factor X or factor 5, phospholipids and calcium form prothrombinase. Prothrombinase then converts prothrombin to
and
thrombin.
begin to stick to the walls of the blood vessel.
Thrombin then converts fibrinogen to fibrin, and then finally the thrombin activates the clotting factors, promoting clot formation and stabilizing the fibrin clot. Clot retraction and fibrinolysis. Okay, clot retraction is the stabilization of the clot by squeezing serum from the fibrin strands. Results from the contraction of platelets, which then pulls the edges of damaged tissue closer together, and then serum, which is plasma minus fibrinogen and some clotting factors, is squeezed out to the clot, followed by thrombin and tissue plasminogen activator activating plasmin, which dissolves fibrin.
The infected tissue.
Thrombin and tissue plasminogen activator. Plasminogen is activated by thrombin and tissue plasminogen activator to form plasma.
Towards the following
okay, and then that plasmin activates fibrin, and then the fibrin breaks down clot in fibrinolysis. Okay, clot or thrombin and tissue plasminogen then activate and convert plasminogen into plasmin in step 1, and then plasmin breaks down fibrin in a clot, resulting in clot fibrinolysis. Okay, blood grouping. We did this lab in class previously, so you should have a pretty good understanding of what the blood types are. Okay, blood grouping. Red blood cell membranes have glycoprotein antigens on their surfaces. These antigens are unique to individuals, are recognized as foreign if transfused into another individual, which is why blood typing is very important before a patient receives donor blood. Okay, promoters of agglutination are referred to as agglutinogens. Okay, presence or absence of these antigens is used to classify blood groupings. Transfusion reactions occur when mismatched blood is infused, and then the antibodies can bind to the donor's red blood cell antigens, resulting in agglutination or hemolysis of the red blood cells, leading to diminished oxygen carrying capacity, clump cells that impede blood flow, and ruptured red blood cells that release free hemoglobin into the bloodstream. That's the main problem right down there. Ruptured red blood cells releasing hemoglobin into the bloodstream. That can have a negative effect on the body greatly with excess iron. Okay, ABO blood grouping. Okay, the ABO blood grouping consists of 2 antigens, A and B, on the red blood cells and in the plasma. Okay, so if you are type AB, you have antigens present for both A and B. Okay, you do not have the antibodies for anti-A or anti-B. Okay, if you're B blood type, you have the antigen for the B, and you have the antibody anti-A. See how that shows up? If you have it, then it's the plus, and if you don't have it, it's the minus. Okay, A. If you have the antigen A, you're you're A. Okay, you see the plus sign where the A is. Okay, there is no anti-A, but there is an anti-B. If you're O, you don't have either antigen, and you have both antibodies, anti-A and anti-B. So let's look at these cells here. We see antigen A for the type A blood cells, antigen B found on type B blood cells, and then you see that type AB has both the antigen A and antigen B on it, whereas type O cells have neither antigen. but have both antibodies. Nice little picture there. It really explains the blood typing well to me, seeing that, before I could see this always made sense. This to me took a long time to figure out. So seeing this in a chart that shows how they work, if you have A but not B, then you have A antigen and the B antibody. If you have B blood, then you have the B antigen and the A antibody. I got them confused a lot. Antigens and antibodies. Remember that antigens are the type. The antibody is against the other one. Okay, agglutination reaction. Okay, type A blood will donate to a type A recipient. Okay, does not cause an agglutination reaction because the anti-B bodies in the recipient do not combine with any A antigens on the red blood cells. Okay, and then we look down at the bottom. If you have agglutination reaction, okay, a type A blood donated to a type B recipient would cause agglutination because the antigen A would match up to the antibodies present in a type B person because they would have antibodies for type A. And this would cause major problems in an individual and rejection of that blood transfusion. Okay, there's a nice little video here. I want you to make sure you watch this one as well. Remember that my videos are not playing properly for some reason, so make sure that you watch this one on your own if you possibly can. Okay, Rh positive blood has certain Rh antigens, including the D antigen, whereas Rh negative bloods do not. Antibodies against the Rh antigen are produced when an Rh negative person is exposed to Rh positive
and release
blood.
The enzymes and
okay, the Rh blood group is responsible for
death.
hemolytic disease of the newborn, and that is why the RhoGAM shot is given to those that don't have the matching correct combination of the Rh positive and Rh negative mother to fetus combination. So if the fetus is Rh positive and the mother is Rh negative, that could be a major problem. Okay, hemolytic disease of the newborn. Okay, well, since the baby is Rh positive, the mother would see it as an invading body, and the mother's system would attack the baby and cause problems for both the mother and the baby. So before or during delivery, the Rh positive red blood cells from the fetus enter the blood of the Rh negative woman through a tear in the placenta. Okay, and then we get to number 2. After the problems have started, the mother is sensitized to the Rh antigens and produces anti-Rh antibodies.
After killing and
because
digesting
this
the
usually
bacteria,
happens
the
after
neutrophil
delivery.
is
There is no effect on the fetus in the first pregnancy. Not a guarantee, but sometimes, or most of the time, it happens during giving birth. During a subsequent pregnancy with Rh positive fetus, the Rh positive red blood cells cross the placenta, enter the maternal circulation, and stimulate the mother to produce antibodies against the Rh antigen. Antibody production is rapid because the mother has sensitized to the Rh antigen. The Rh antibodies from the mother cross the placenta, causing agglutination and hemolysis of the red blood cells of the fetus, and hemolytic disease of the newborn develops.
In size,
diagnostic
and
blood
into
tests.
Smaller pieces.
Blood tests are very easy nowadays. Laboratory examination of blood can assess an individual's state of health. Microscopic examination. Variation in size and shape of the red blood cells
can be recognized
can predict anemia,
and eaten
especially
by
sickle
other
cell
cells.
anemia, because they show up flashing bright red, basically. You can tell by the shape of the cells that a person has sickle cell anemia. Type and number of white blood cells, the diagnostics of various diseases and infections. Chemical analysis can then provide a comprehensive picture of one's general health status based on relations to normal values and concentrations in the blood. The complete blood count consists of the following: the red blood cell count, which in males is between 4.6 to 6.2 million per microliter, and in females is between 4.2 to 5.4 million per microliter. Hemoglobin measurement, grams of hemoglobin per milliliter of blood.
Cell is the functional
Males have
basic
14 to
unit of
18
life,
grams
often
of
called
hemoglobin per 100 milliliters. Females have 12 to 16 grams of
building
hemoglobin
block of
per
life.
100 milliliters.
Humans have about
Hematocrit measurement.
100
Okay,
trillion
this is the percent volume of the red blood cells. Male have a hematocrit percentage
cells.
of around 40
Your
to
body
52%,
is constantly
where females'
producing new cells.
hematocrit is 38 to 48%. So males typically have more cells in their blood. Okay, white blood cell count. White blood cell
In
count
this
per
program,
microliters.
we will
Male and
discuss how
females
cells
both have
divide,
between
how
5,000 and
chemotherapy affects cells
9,000
that are
white
divided.
blood cells per microliter. Okay, the differential white blood cell percentages. Neutrophils are the
and
largest
how chemotherapy
of the
affects
group
cancer
as far
cells.
as numbers, 60 to 70%, followed by the lymphocytes, which make up 20 to 25%, followed by the monocytes at 3 to 8, eosinophils at 2 to 4, and then finally your smallest portion, your basophils.
When
at
a normal
0.5
cell
to
divides,
1%.
it goes through
Okay. So a way you can find out the hematocrit would be to centrifuge the blood down. As you
a
centrifuge
series
the
of
blood
steps,
down,
including a duplicate.
the red blood cells will go to the bottom. The white cells and the platelets will form an area in between the red and the plasma, and the plasma will make up the largest percentage of the
of
blood.
its DNA,
Okay,
which
here in the
contains the
hematocrit
genetic
tube.
instructions used in the development.
Okay, clotting. Platelet count and thrombin, prothrombin, time measure the ability of the blood to clot, and the blood chemistry. The composition of
functioning of
materials
living
dissolved or
organisms.
suspended in
The end
plasma can
result
be
of this-
used to assess the functioning and status of the body systems, including glucose, urea, nitrogen, bilirubin, and
Cell
cholesterol.
division is the formation of two identical daughter
All right.
cells.
Remember, keep check on Course Connect. Course Connect has a couple things added there for you. One, the test will be posted. It's
Let's
actually
begin
already
to
been
explore how
created,
this
and it will
process
be
happens.
posted for Thursday. Let me see what date that is.
The cell goes
Really
through
quick.
many distinct steps as it divides.
Okay, Thursday the 24th. Okay, you'll have several days after that date to begin that test and get it completed. Please make sure that you have it done on time to get the best and most accurate results and the quickest response. I also have it set this time that you will get the rough score
After a cell's
as
initial
soon as you
formation,
finish your test.
it
You won't
goes
have
into
the
an
final
inactive
adjusted score based off
resting
of
phase
the
called
other factors that
G0.
work into it, such as any bonus or anything like that that goes into the score until after
The cell
the
then
test
grows
is
larger
officially
in
over
size
with and
and
anything
makes
else like that gets
proteins.
factored in. All right. So let's go ahead and end this lecture. And you guys have a
These are
great
important
week.
for the
And